Dr. Lum, it's Dr. Julian Lum who's going to speak to us about prostate cancer research update. Dr. Lum was born and raised in Ottawa, Ontario, and graduated with a PhD in immunology at the University of Ottawa. He spent 5 years training at the University of Pennsylvania in the field of cancer biology. In 2008, Dr. Lum returned to Canada and joined BC Cancer, where he started a research program to understand how the immune system responds to prostate cancer. His team is currently conducting studies to combine new forms of radiation therapy with immunotherapy for the treatment of late-stage hormone refractory metastatic cancer. Long line. Dr. Lum is an associate professor in the Department of Biochemistry and Microbiology at the University of Victoria, and is a senior scientist at BC Cancer, just down the road. He was previously the recipient of a prestigious Canadian Institutes of Health Research New Investigator Award. So join me in welcoming Dr. Lum.
Okay, fantastic. Thank you very much for the kind introduction and coming out this evening and spending some time with us and with me. So I wanted to start off just by saying that I've actually been one of the more fortunate ones in terms of knowing people in my immediate family who have had an experience with cancer. Not very many people actually in my family have, but my wife, my father-in-law, my wife's dad, just was recently diagnosed with prostate cancer. And so we're beginning that journey with him. He actually hasn't even yet seen his oncologist. But that's something, that's very near and dear to my heart. And of course, the work that I do in my research lab really grounds me in the kind of things that I hope to do and hope to at least bring some hope and ideas to how we can treat this disease. Okay, so thank you for the introduction and telling the folks a little bit about me. I also wanted to just start off by telling you a little bit of a story of how I got into prostate cancer research, or cancer research in general. When I was growing up, I was an avid fisherman. And I was also an avid fish keeper. So we had, at one point, 8 aquariums in our house of all kinds of different varieties of fish. And so, many of you know, who have had pets, fish or otherwise, that invariably they get sick. And so for me, my first experience with this was with my fish. They got sick, and I didn't know what to do. I was kind of lost. I went to the store. I asked the people there. I described to them the symptoms. I said, the fish have these little dots on them, and they look like, I don't know, bacteria or something. I don't know. And the person at the store said, well, I know what your fish has. Here's the medication. Why don't you take this home and do this treatment? And I said, sure, great, that sounds fantastic. So I did that, and about 2 weeks later, something surprising to me happened. So some of the fish got better, and they were fine. These spots disappeared, but some of them died. And that was my first experience with keeping an aquarium where the fish died. But I really got curious about that. I said, they're all in the same tank. They all had that same white speckling on them. I gave them the same medication that the person at the fish store told me to give them, but yet some of them died. So why is that? And I think that really became kind of a catalyst for me to begin my sort of inquisitive, scientific endeavor. And so I went on, to university with that kind of in mind. Okay, so I'm going to share with you today some of the things that we've been doing at BC Cancer in terms of treatment. Of course, we do many more things than simply treatment. And this first slide here is really the analogy I want to to use, which is that cancer is like that orange clownfish, okay? And as you know, for those who know something about ornamental fish, clownfish exist in a symbiotic relationship with sea anemones, right? And they hide in the sea anemones from predators. And the sea anemones protect them because sea anemones can secrete poisons, right, that fend off prey. And so I think that's a very good analogy because cancer or cancer cells are cells of your own body. And they're hiding. They're hiding amongst all the normal cells that are found in the prostate. And because I'm going to talk to you a little bit about the immune system has to figure out who's the good guy and who's the bad guy amongst all these different cells. Okay? So hopefully that will set up what I'm going to talk to you about. I want to acknowledge lots of people. I'm not going to go through all their names, but this is really a major, massive team effort. From people in my own laboratory, to the medical oncologist, to the radiation oncologist, some past and present members of BC Cancer, you can probably recognize some names, our colleagues in Vancouver as well, okay? And funding from a large variety of sources that enable the work that we do. I'll very quickly go through this because some of you probably know BC Cancer is a provincial entity. We are sort of the one-stop shop in the province for all cancer patients to come, receive their treatments. We have different sites, of course, scattered across the province. And the one thing I'll point out is that our main research hub, of course, is located in Vancouver, but we do have a research arm of BC Cancer here in Victoria. Okay, we're on the 3rd floor of the Cancer Center, and some of you may have seen us if you've been through the building, and I'm going to tell you a little bit more about what we do there. And we have links to some of the academic institutions, both locally and across the province. So what I really want to talk to you is about new opportunities, new things that we are working on that hopefully we can bring those to the clinic to basically treat patients. And we focused at this time on patients with very late stage disease, the hormone refractory metastatic disease. And it's really about ways that you can harness your own immune system to fight cancer. And I think that is the difference in terms of what we're trying to achieve with respect to other avenues of research, such as those that are looking at new forms of chemotherapy or new forms of radiation. So the immune system forms a new way to think about things. This quote here, I think, really sums up why it's such a complex disease. And I'll just read it to you. It says, "No two people are alike, including their response," and I usually like to put the word immune response to cancer. And the more we know why, the better we can treat people as individuals. Each and every one of you sitting in the audience tonight, we're different. Our bodies are different, our immune systems are different. If you're a man and you have prostate cancer, your prostate cancer is different from the individual sitting next to you that might have prostate cancer. And I would extend that to women's cancers as well, okay? So we really need to understand the individual in order to be able to tailor a treatment for that cancer, for that individual. And that's what we do in the lab. We do lots of very complicated things, and then something very magical happens, okay? And then out comes some new treatment, okay? And what I hope to do is demystify a little bit about this middle part that we're working in the lab and we're doing all these interesting things, but what exactly are we doing? Okay, so just as an outline, I'm going to talk a little bit about the current prostate cancer treatments and the immune system. I'm going to talk to you about new types of what we call immune therapies. Immune therapies are ways that we stimulate or reprogram the immune system to direct its attack at the cancer. And we're going to talk a little bit about how we can personalize this, right? Individualize it so that it works for that individual and it's precise in its attack. Okay, so some of the standard therapies, many of you know, but I'm just going to very quickly review this. So prostate cancer is what we call a hormone or androgen-sensitive cancer. And to put it into a diagrammatic form, imagine this circle here. This is a cancer cell, a prostate cancer cell. This cell depends on different kinds of androgens in the environment. Okay? And what happens is those androgens get taken up by the cell, by the cancer cell, and that gives that cancer cell an instruction. Okay? It tells that cancer cell it's time to grow, it's time to divide, it's time to make more of the cancer cell. Okay? So that's what it does. But the one big issue is that the cancer cell, by definition, has a mutation. It's genetically rewired differently than normal cells. Right? And mutations occur in this molecule, which we call the androgen receptor, that no longer requires those androgens as the signal to grow and divide. Okay? So this cancer cell can grow even in the absence of those androgens. That's the stage where we call hormone refractory or androgen independence. They don't need it. The drugs that are used to treat that what was a sensitive disease is no longer active, is no longer an active drug. That's what we call hormone refractory, sometimes referred to as castrate resistant.
Like the first term, better actually.
Agreed. And feel free, please, to interrupt if you have questions, okay? So the treatments that we have in the clinic are, we block the actual production of that androgen. We block the ability of the androgen to enter into the cell, and we can block the ability of the androgen to bind to the receptor. Those are the three main hormone treatments that we have, that we deploy in the clinic right now. Okay? And the end result of that is to inhibit or block that cancer cell from growing and dividing. In conjunction with hormone therapy, there's radiation therapy. Okay? The most common form is what we call external beam. So this is a machine that generates ionizing radiation, and we can direct a beam of radiation very precisely to the cancer cell. Okay? Brachytherapy, some of you may have heard. Okay? These are essentially little seeds, little pellets. They're radioactive, that are surgically implanted into the cancers and into the prostate where the cancers reside, and they give off the radiation right where these little seeds have been deposited. That's called brachytherapy. Okay? And both of these have been highly effective, along with and in combination with hormone therapy. But many people, many men, will progress to this hormone-resistant or refractory stage. And we need new treatments to be able to get at that stage of disease. So does anybody know, or can you recognize this fellow in the...
He's a Nobel Prize winner.
That's a very good guess. Very good guess. He is not a Nobel Prize winner. He should be, I think, but he's not. This is Martin Cooper. And Martin Cooper worked for Motorola. And he was the one who invented the very first, at the time, what we called the mobile phone. very large, handsets, right? Okay? But that was a big, big change in the way that we communicate at that time, right? We could now walk around with this large device and talk to our peoples, our friends, and so forth. This man needs no introduction.
He treated his own cancer.
He fundamentally changed the way that we communicate. Completely different way that we think about how we talk to people, how we interact with people. I go up to UVic now when I give lectures, and every student is in the classroom too, right, while I'm giving my lecture. So it's a very fundamentally different way to think about, at the time when Steve Jobs came up with this iPhone, a very different way to think about communication. And I think that's what we need. We need a fundamentally different way to treat and attack the problem of prostate cancer, okay? And we need a different approach to doing this. So, traditional cancer therapies, like the ones I talked to you about, systemic chemotherapy, even radiation, surgery is part of that mixture. They kill the cancer cells, we know that. But they also kill healthy cells. There's a side effect of these drugs. Excuse me. And immunotherapies, those therapies that are stimulating your immune system, okay? They are very selective. They kill only the cancer cells, and they spare the healthy cells. Right? And we know that because the immune system has exquisite specificity, meaning that it recognizes when you have a cold, a virus, it recognizes if you have a bacteria infection, and it only kills the bacterial infected cell. It doesn't kill your normal cells. And then once you resolve your infection, your immune system goes back to a silent state, right? Now there are exceptions to that, because if you tilt that balance too hard, your immune system can turn on your own tissue. That's called autoimmunity, right? Okay, but there is a very high degree and high level of specificity that the immune system maintains, for the most part, in most people, this very healthy balance. Okay?
Does that change with age?
That's a very good question. So, as you age, you become less able to combat things like flu or other viruses, because your immune system has aged as you've aged, and there's an effect whereby the young immune cells that you've had have died off, and you're no longer able to replenish that in a way that's similar like my 8-year-olds can, right? So, does that answer your question?
Yeah.
So what is the immune system? The immune system is a network, a connected network of arteries and veins that the lymphatics allow your white blood cells and your red blood cells to travel along. And within those white blood cells, there are an important group of cells that are the soldiers, if you will, that are the ones that are going out and looking for bad things. Those cells are called T cells, okay? And T cells, in order to be able to destroy a cancer cell, they need to be physically in contact with them, because these T cells latch on to the cancer cell and they actually inject poisons, biological poisons, into the cancer cell to kill it, okay? And it will only attack the cancer cell if you tell it to, and not the normal, healthy prostate tissue or prostate cells. So we call these T cells the soldiers of the immune system, okay? And this is the cell that we're going to focus on in the next part of my talk. So there is now a type of treatment whereby we can take a blood sample from a person that has cancer. This has been done in prostate cancer. We can take a blood sample. We can extract the T cells, right? So we can find these T cells that are the ones that are directed against the cancer cell. And we can modify them in the laboratory using genetic approaches. And we modify them to make them better at recognizing the cancer cell and finding the cancer cell and injecting those poisons, attacking and killing the cancer cell. Okay? So we can do that. And then what we do is that we grow those cells that we've modified in the laboratory into billions and billions of these cells. And very similar to a stem cell transplant, we infuse back into the bloodstream those modified cells, which we've made better, those T cells, those soldiers. We infuse them back into the cancer patient that we got those cells from, right? And the idea is that those cells are an army, a wave of army that just goes into this body, finds the cancer, and destroys it. We call that CAR T-cell therapy. And the reason why we call it a CAR cell therapy is for the following reason. So you extract from the blood the T cell, and you put a CAR. It's a molecule. It's like a little beacon. And that beacon gives this soldier cell, this T cell, direction, just as the name implies. Okay, it's going to find the cancer cell, and once it finds it, it's going to kill that cancer cell. Okay? So the product of this is called the CAR T cell. Okay? We are very fortunate because here in Victoria, we are one of maybe 3 centers in Canada and one of maybe 15 centers in the world that can that has the ability to do this. We have the technology and we have a facility that we can produce and make these CAR T cells for patients. This is the 3rd floor in one of the research labs, so just right over here at Kitty Corner at the Cancer Center. And it's called the Conconi Family Immunotherapy Lab. This photo here, these 4 individuals, these are the Conconis, the family. This is Dr. Brad Nelson. Some of you may have seen or heard of him. He's the director of our cancer center. And this is Sarah Roth. She is the president and CEO of our foundation. And we were very fortunate enough that the Conconi family pledged some dollars to help us build this facility. And again, we are now capable of making these, what we call clinical grade CAR T cells. Okay? This is Victoria. She's one of the staff members that work there. And she's working in one of these specialized incubators. It's called a cell processing unit. So you can see she's behind a glass where she's got her gloved hands inside the box. Everything inside this area is highly sterile, right? So that the product she makes that we're going to infuse back into a patient is clean and meets regulatory standards. Okay? And so CAR T cells, this is really just the tip of the iceberg. Okay? And this is our first generation, our first pass at attempting to do this. And so I'll use some specific examples now. In the world, as I said, there's only about 12 or 15 places that can do this. And one place right now in Memorial Sloan Kettering, which is in New York City, They have a clinical trial where they have put a CAR that specifically recognizes prostate cancer. Here in Victoria, we are using that same framework, that same backbone, that CAR, but we're modifying that CAR to be even more potent at killing the cancer cells. So we have what I would call a second generation CAR. Okay? We are still in early stages of developing this. I'm going to show you in a few minutes how long it kind of takes and what it takes to get from product all the way to clinical trials. Okay? And our hope is that in the next 100 years, we're going to be having supercharged cars, right? Ones that we can individualize, one that we can ensure that they're directly attack and wipe out all the cancer cells. Okay? Okay, so I'm going to stop here, and I'm going to switch gears a little bit, because one of the things that I think that a lot of people ask me when I'm giving some public seminars is, well, we've heard about this or that several years ago. What's happened to that idea? Why can we find the drug that was made from that, or where are we at? How come we haven't started patient trials? So I want to tell you a little bit about what it takes, the time it takes to get from idea all the way to being actually trying this in patients. And I also do want to touch on a little bit of funding, because it's something that I think it is surprising to a lot of people, what it takes to go from just an idea all the way to a product. So I spend a lot of time fishing. Well, I used to before I had kids. I think you probably all can appreciate that. But when I do get a chance to go out, it's a great place, because I get to think. It's quiet most of the time. But I get these interesting ideas while I'm fishing. And it's a moment where I think, oh my God, why haven't we done that? This is an amazing idea. We have to try this. So I have this sudden moment. I have an idea. Now what? Well, I need to test that idea. I need to figure out if that idea actually works. So there's something called the preclinical stage of research. And that preclinical stage, on the order of about 5 years. So just putting pen to paper and saying, I have this very interesting idea, this is what I want to do, and now I have to execute that. I have to go into the lab and test that idea. That costs upwards of $500,000, so about $100,000 a year just to test the idea. And I'm not that smart, because 9 out of 10 of my ideas fail. And maybe one of those ideas will go on to the next stage. Hooray, that next stage, we've made it. We've now taken an idea and given it some backbone and some legs, and that idea now we can move forward. So we go to the next stage, which is called preclinical trial stage. And the preclinical trial stage is a stage where we need to seek Health Canada approval to do the first in-person study. Right? It's a brand new drug. We're going to try it in a human being. That requires a lot of regulatory approval. Okay? That can take up to 2 years, and that can take up to $250,000 to do that. So if you're keeping track of the dollars and cents now, we're at about $750,000 and about 7 years worth of work. More or less, plus or minus. If we pass this stage, we get to do something called a phase 1 clinical trial. And that phase 1 clinical trial, that trial asks one question only, and that question is the drug safe? If I give this drug, will that cause some side effect that we could not anticipate in the lab. They're typically done in very, very late stage patients, ones that have tried every possible therapy, and there's really not very many options left, or if any options left. It's done in a small number of patients, and the cost is around $650,000 just to do that. Usually about 10 to 15 patients, okay? It takes about 1 to 2 years to do that. Okay, so keep track now. We're adding not only years, but dollars. If this is successful, meaning your drug is safe, it doesn't cause some undue reaction, you get to go to the next stage. And that stage is called phase 2 clinical trial. This stage asks another very simple question, and that question is your drug effective? Meaning, can your drug cause some change in the trajectory of that individual's cancer? Usually between 50 to 100 patients. It's about a $2 million effort. And it takes about 1 to 2 years, conservatively, to do phase 2. Okay? Don't forget, at each one of these stages, the chance of failure is very high. Your drug is now effective. You need to do a last phase 3 clinical trial. And the phase 3 clinical trial asks one more very simple question. Is your drug, which you said was effective, is it better or equal to the standard of care? What is already prescribed in the clinic that the patient would typically receive? Okay? It's a very large study. Oftentimes more than 300 patients. It's multi-institutional. It's a $20 million endeavor. And it can take anywhere from 2 to 4 years to complete this study. Okay, so some of the hormone therapies, the new ones that have now been approved for use, for example, enzalutamide, maybe some of you have heard of that drug, or abiraterone, these are the hormone blockers. They're sort of the next generation hormone blockers. Those drugs went through this entire process. Right? So if we don't consider this part of the equation, the development of a new therapy can take anywhere from 9 to 14 years. And without considering these clinical costs, I'm just talking about here to here. We're on average about $1 million. So, I think a lot of times when I talk to people and I tell them, well, this is a long-term investment, and many of the ideas that we try do fail. That is the nature of our game. And I can remember 4 years ago when I gave a talk here, we were onto something really exciting, and we were actually in sort of that early phase 1 trial. We had a drug company that was interested, but it turned out, unfortunately, that the drug was not as safe as we thought it would be. So we've gone back to the drawing board with that. But now you see we went backwards, not forwards, and it's a long process. Is really what I want to convey. How do we fund our research? This is a very important question. So the Canadian government, the federal government, has a research funding arm called the Canadian Institutes of Health. They fund about 300 to 400 research grants per year across all health disciplines, not just cancer, across every health discipline. Our dollars are used to pay for salaries, students, that takes about 60%, our consumable costs about 30%, and then we have some other costs such as traveling to conferences, talking to our colleagues about their research successes and their research things that have worked for them and not worked for them. Funding that unfortunately doesn't allow us to purchase equipment. And that's a big gap in the research enterprise right now. Okay, so I'm going to just go back a little bit here. I hope I meant to bring this from the laboratory, but I was delayed because of 2 little children. This little test tube here that I'm circling in blue, it's got a blue cap, and it's basically a big test tube. It's about this big, about this wide, and this is Victoria, and she has to use these every day. And you can see here she's got some Petri dishes, and she's got some bottles, and a few other things in here. But I just want to ask the audience, how much do you think one of these little it's made of plastic. How much do you think one of those little plastic test tubes cost? I should have brought one, but I just ran out of time before coming.
So, what it costs for you to make?
To buy one.
Oh, to buy one. Well, the plastic itself, I don't think it would be too expensive, but the inside, the process will be at least $100,000.
Okay. So the plastic is not very expensive, but the insides are very expensive, and I agree with that.
The T cells.
Yes. So how much do you think that plastic costs? Let's see if there's something. Yeah, it's actually about this big. Made of plastic. Clear plastic with a blue cap on it.
49 cents. $1, $2.
Who thinks more than $10? Raise your hand. Who thinks less than $1? Raise your hand. Okay. So here's the exercise we're going to go through. Here it is. A 50 milliliter test tube. One test tube costs 25 cents. So you might be thinking, oh, that's not so bad, 25 cents. On a typical day, one person like Victoria, who's working in that specialized cabinet, she uses about 20 of these test tubes. That's not so bad, that's $5. There's about 55, 60 staff, students that work over at our cancer research center now. And I know all of them are working in the lab all the time, 24 hours a day. But let us assume that only 10 of them are working on that day. So if 10 of them use 20 test tubes at 25 cents per day, it's about $50 for 10 people. That's a very conservative estimate. So $50, I know they work on weekends, but let's just say they work 5 days a week. They like to take holidays. They like to take holidays so we don't work the full year. So we work 45 out of the 52 weeks. That's how much it costs per year, just for that one little test tube, for 10 people to do 20 experiments a day. And that is a very conservative estimate because we have 60 people working in the lab at any given time.
Why don't you wash them and reuse?
Very good question. Yes. We can't because when you're working with one person's cells or one person's tumor, you can't contaminate it with another person's cells and another person's tumor. Those, I really wish I brought them, but those tubes are sterilized so that you only use them once. Okay?
Can the process be automated?
Can the process be automated? Are there any engineers in the house? If you can automate this, that would make me very happy, but make my graduate students very sad because they wouldn't have a job. But I think there's some hybrid. You know, you can do something in between. There are certain things that I think we can automate. And you know, I consult and visit large pharmaceutical companies, and I tell you, they have rooms and rooms 3 times the size of this auditorium with robots. And it's just all done in automation. But the cost of that is just not academically possible. I mean, with in an industry, with investments and so on, that's in the realm of possibilities. I'm going to give you one more example. This little bottle here, it's about that big. Okay, I meant to bring one. Try to find some. Well, it's about that big actually. It's a little longer, not as fat, but it's about that big. And inside that little tube is a special reagent for doing an antibody test. Okay? And there's about 1 dropper worth of liquid. So your eyedroppers that you use, there's about 1 drop of liquid in that. Okay? We can do about 100 to 150 tests. And on average, we do about 50 tests per experiment. So each time we do an experiment, we use 50 tests. We do 50 tests. So how much do you think this bottle with 1 drop of liquid in it costs?
Because it's made by the pharmaceutical companies, that was $3,000.
You're pretty close, not that far off.
Okay, 999.
So typically we do 50 tests. Each year we use about 10 to 15 of them. Okay? One of these bottles costs on average about $450. So I won't do that math, but what I will show you is this picture. So I went into the lab and I said to my students, can you show me what we have and how many of these little bottles we have? So they took out, this is about half of what we have. Each one of these bottles is about $450. We have 1, 2, 3, 4, 5, 6-ish. That's half of what we have, so we have about 12 of these boxes full. So it really is better to rob a bank, or better to rob a research lab than a bank, because there's a lot of investment put into this. But I think what I really wanted to get across to you is the scope of what it takes for us to do the research that we do, and the time that it takes to get from the idea all the way to actually saying, okay, now let's treat patients. Okay, so I'm going to stop here. I want to thank a lot of different people. This group here has been hugely supportive of the work that we've done in the past, currently, and I'm sure in the future. I'm happy to answer questions now, and thank you all for coming this evening. Okay. We'll start at the back if there's a question. Yeah.
I was just wondering if you could tell me whereabouts you are now, like what phase you're at with your CAR T cells research.
Very good.
How far you have to go until you get to the last one.
Yeah. Excellent question. So we are the first place in Canada that's going to deploy and test CAR T cells. We are using a very different platform than what is being used in the US and used in the UK and other jurisdictions. So we need to go through a large series of vigorous regulatory steps to show that our platform, the way that we make those CAR T cells, is safe and equivalent. So right now, there are 2 CAR T cells that are already approved for use in acute myeloid leukemias. These are blood cancers. They're made in the US. The sticker price right now to enroll, to get access to this in the US, before they even talk about, making the cells for you, is about $475,000 US. So here in Canada, we think we can do it cheaper, better, much more cost effective within the context of a national healthcare system. So we have now the first generation Canadian-made CAR that we're going to first test in leukemias as a biosimilar, meaning it is a product that is similar to the one that's being made in the US with some differences, because there's obviously a lot of patent and intellectual property that you have to consider when you do these sorts of things. We are partnering with a couple of different institutions across Canada. The Ottawa Research Hospital is one of the partnering institutions. Toronto will be a second one later on. We have received the regulatory approval, i.e. sort of that first step. We are hoping to enroll the first patient within the next couple of months to receive this therapy. So the patient will be treated in Ottawa, but their cells and the generation of that CAR T cells is going to happen right across the street. And then we're going to ship those cells back to Ottawa, and the group in Ottawa is going to do the infusions. Yeah, go ahead.
Just to follow up,
Yeah.
that isn't for prostate cancer. Are you doing anything with
Yeah.
CAR T cells for prostate cancer?
Yeah, so my laboratory specifically, we are in the phase before we're going to go to that regulatory step. That's where we're at now. So if I can just back up and just remind everybody, we are right here. We've got the success in the lab. We are now just in this space where we're getting ready to prepare the documentation and everything to seek regulatory approval.
Does success in the lab mean animal tests?
In some cases, yes, if it's appropriate. In other cases, no, if it's not appropriate. It depends, yeah. Yes?
I've lived in a lot of cities in the world, and everywhere I go, there's basically a cancer center, and it's a terrible disease. Everybody's working on it, and I just want your comments. Is there enough coordination so that you're not working on the same thing that they're working on in Winnipeg and they're working on in San Francisco? Or is there some overall general who says, you work on that and you work on this, so we have to focus on this horrible condition?
Yeah, fantastic question. And so there are two ways that we try and make sure that we're not duplicating effort. The first way, as an academic, we publish our findings in journals, and everyone in the scientific community can access and see, oh, okay, they've done this, so we're either not going to try this or we're going to improve on this. The other way to do it is to attend conferences. And we talk to our colleagues. What are you doing in this area? Prostate, whatever. What have you done? What haven't you done? How can we work together? So this first CAR T cell trial is a collaboration between multiple institutions in Canada. And we've worked together to make sure that, you know, within the Canadian system we can deploy this. So that's a very important thing, because the last thing you want to do is to duplicate effort. There's a question here.
My question never get answered.
Oh, it did. Okay, at the back. Yeah.
I think one of your peer group was here about a couple of years ago, and he was talking about focusing on similar things to do with breast cancer. So does a one CAR T cell attack different cancers? Or is it specific to prostate cancer or breast cancer or colon cancer or?
Very good question. So the short answer is it's very individualized to the cancer and to the person. To the cancer, because the CAR, the beacon that we put on, has to recognize just the prostate cancer and not some other cell or some other cancer. Maybe it might be an individual have two different cancers, right? They're a bit more rare, but that does happen. So it's individualized from that perspective. It's also individualized because
that person is that disease.
Correct.
But not to that person and cancer.
Well, it is to that person and its cancer as well, because the T cell comes from that patient. where you put on the car.
Let me toss out an example. Let's say a person has prostate cancer and they also have colon cancer.
Yeah.
Would it only work on the prostate cancer, or would it only work on the colon cancer, or would it work on both?
You would need to make 2 different CAR T cells, one that would recognize the colon cancer and one that would recognize the prostate cancer.
Okay.
Yeah. Good question. Yeah.
I was wondering then, do you need to have different CAR cells for 2 individuals, both with prostate cancer? How individualized is it relative to one type of cancer?
So the holy grail would be you would have a bank of cells that are already made up, and you can call up the one CAR cell that you've made and give it to an unrelated person. That is the holy grail, but we're not there yet. And so with our current technology and the current understanding of how we can produce these CAR T cells, it has to be for that individual, for that cancer.
So how do you test that clinically then in other people?
Yeah, that's a great question. So I'll use prostate cancer as an example because that's one CAR T cell area that we're working on. Prostate cancer cells on the surface of the cell, they have a protein, a molecule called PSMA, not PSA, PSMA. Not all of the cancer cells from individual A or individual B or C will have that PSMA, but many do. So the CAR that we're designing is directing the T cell to the PSMA molecule on the cancer cell. So we can use the same CAR for prostate cancer. We couldn't use it for breast cancer or colon cancer or another cancer, but we can use the same CAR for a person whose cancer has that PSMA decorated on its surface. So from that point of view, we can make one CAR. We still have to put it into the person's individual T cell, but we can make one CAR that could potentially treat multiple different people with prostate cancer, but their T cells are individualized, so we still have to make it for every individual. Right?
Yeah.
So,
That's not very promising.
so this part is the individualized part. This part is the CAR that targets PSMA. So we can put that CAR on one patient's T cell. The next patient comes in, we can put that same CAR on that person's T cell. They're still recognizing that PSMA, but this has to be
Yeah,
individualized.
understood.
Yeah.
Thank you.
Okay. Over here and then over here. Yeah.
My question is a little different, but I'm really curious, given the fact that every molecule in the body is there for some sort of purpose, how is it that cancer cells, which we're born with in our bodies, what is the function or original purpose that those cells are inside of us? Because at this point, it seems like they are an enemy cell. So what are they doing there to begin with?
Mm-hmm. Great question. So this is an evolutionary problem and an evolutionary question, right? And I go back to my clownfish analogy, okay? Their cells, the cancer cells, are our own cells. They weren't transplanted from another individual. Cancer is a disease of genetic mutations. And those genetic mutations are either passed along from your parents and your grandparents and your great-grandparents, etc., or those mutations arise due to environmental exposures. Asbestos for things like mesothelioma, sun for skin cancers. There are viruses that cause cancer, human papillomavirus. But still, the cell is your own cell. And so Why are they there? Why have they become that way? I have a sort of a thought on this. I don't know if it's right because I don't have any empirical evidence that that's my thought is correct. But there's supporting evidence from others in the field that cancer cells rely on food to grow, just like every other cell in your body. Cancer was never a problem or an epidemic problem of this proportion that we have now many decades ago, right? The big killers many decades ago were what?
Smoking.
Smoking, heart disease, cardiovascular diseases, etc.
TV.
Yeah, infectious diseases. As the population life expectancy has gotten longer, as we have changed the way that we eat by going to our refrigerators and eating the wrong kinds of things that are fueling these cancer cells, that has contributed to why these cancer cells have become more cancer as a disease, become more not relevant is not the word, but prevalent. Thank you very much. Prevalent. And if you look carefully at when diabetes became a really major health problem, it coincides with when cancer started to become a problem. And there are links between those two phenomena, but it's not perfect, meaning it's not a direct cause and effect relationship. It could be, but we have not scientifically proved that that's a direct cause and effect relationship. But the striking thing is that diabetes and cancer, when diabetes was a big problem, we started to see cancer start to become a problem. Because it's the foods that fuel the cancer cells.
When was that?
Late 60s, early 70s, I would say. There was the spike in sort of the diabetic problem. Got insulin.
Fast food generation.
Fast food generation. Yeah. So, very good question. I don't have the answer, but that's where the science is kind of taking us in terms of our thinking.
Yeah.
So the long-term vision is if you're diagnosed with prostate cancer, you go and you get some tests, you get blood tests, then you get a transfusion with the engineered cells, and then you're all done in a matter of months. Is that the idea?
Yeah, so the whole treatment is actually very short. It takes about 2.5 to 3 weeks to make the CAR T cell and to get billions of them. The infusion is maybe an hour long, and you're in the hospital and you've got an IV, and then the time it takes then to figure out if it works takes a bit longer. And so the idea is that because they're your own cells, right? It's like if you get an infection, your immune system responds to it. There's a ginormous spike in those T cells. They wipe out the infection, and then your T cells go into this, what we call a memory state. It's like vaccinating yourself. So the next time that you get that same infection, your immune system is primed and ready to go. It'll see that, and it'll likely eliminate that infection before you even get symptoms. And the same idea and the same principle applies to these CAR T cells. The cancer's there, you infuse the CAR T cells destroy the cancer, they sit in your body and they wait, and if there's another cancer cell that pops up because it didn't get all of it, those T cells will get awakened again, just like it's like an auto vaccination in your own body.
What are you going to do with all the oncologists and urologists out there?
That's a different problem altogether.
Once, once you have this process perfected, will it be affordable, do you think, or will it be prohibitively expensive?
There are some very smart people in health economics that are grappling with this problem. And I don't have the answer to that. What I can say is that Novartis is the company that makes the CAR T-cell for leukemia. It's approved in the United States. They've come to Canada and they say, we want it approved by Health Canada. They got the approval. But because we're a nationalized healthcare system and we have provinces that now negotiate the price of drugs from pharmaceutical companies as a collective, we leverage that, right? It's not like in the US where you have some HMO here, and they all negotiate their own price. We have a Canadianized system. What's very interesting is that that body that negotiates the pricing between the drug companies and the provinces have said no. Health Canada says it's a safe drug, it can be used, but the province, that body that negotiates that pricing with them has said we will not approve the provinces to pay for this until the drug companies reduce the cost of that drug by half, and then we figure out how to nationally deploy it. So we think making it here, we're the only group in Canada that can make it. So we think here, by our estimates, we can do better than half of what the cost is that this body is asking for. If that's sustainable, that time will tell, and things, the landscape in health, economics might change. So, but we think we can do a much better job. Yeah. Here, and here.
This is a very insidious disease, and I'm just wondering if this thing will go the same route as hormone therapy. When hormone therapy was discovered, it was going to wipe out cancer. And you take hormone therapy, you wipe out all of them, except a few cells always survive. Is there a chance that this too, you're going to develop the T cell, and it'll do 99% of them, but it'll always leave a few, and it just evolves all the time. It's so hard to wrap your head around. Is there a chance that this could happen to this as well?
Yes.
It will work them all out, yeah.
And this is the reason why we think this. Think about, just ignore the CAR cell for a moment. Okay, so you have a person doesn't have cancer. And as they age, some cells have been exposed to things and become cancerous. The immune system is actively trying to rid those cancer cells, and they do. So they establish a little bit of an equilibrium. And then at some point, the cancer cells figured a way to evade and trick the immune system, and now the cancer cell props up, you've got disease, and now you've got to deal with it in a different way. You still have an immune system. It's not like the immune system had gone away, right? It can still respond to bacteria and viruses. So the way that I see it is kind of an arms race, right? The cancer cell is always trying to get an upper hand, and the immune system is trying to always suppress that. But at some point, the cancer cell mutates enough that the immune system can't see it anymore. It's like the clownfish. It's there, but can't see it. And I think the same thing could potentially happen, and may happen, with an approach like CAR T cells. There's no magic bullet, I think, is what I'm trying to get across. But what we hope is that we will reduce the side effects that other therapies might have, chemotherapies, radiation, hormone therapies, because you're using your own immune system to deal with that. So you might, let's say you got a PSMA CAR T cell therapy, your cancer's gone away, and then 5 years later you have a recurrence. We'll test that cancer, we'll see what it has, we'll find the T cells that recognize it, and we'll repeat that whole step again. I think up here and then at the back.
I was going to ask, is there a patent problem for the process? If somebody already has
Yeah, are there
something
any lawyers
similar?
here? We need a lot of help. And because the landscape of the CAR T-cell world, scientific world, is highly complex now. And drug companies got wind of this. Like, drug companies traditionally and historically make drugs. They make, pills. This is not a pill. This is what I call a living therapy, a living drug. It sells. And pharmaceutical companies, in the old days, when they found their pill, they would patent everything around it, right? Every way that you can get to their formulation, they put, blanket patents. And the issue right now is that drug companies have got wind of CAR cells, they're making them, and they're using that same approach. They're just blanketing the entire landscape. It makes it very challenging for guys in the academic world like myself to try and penetrate into that. But we have ways that we can do that. And we are doing things that are different from what the pharmaceutical companies are doing. So I think that's the really nice thing with academics, that we not to say that pharmaceutical companies don't have smart people, but that's what we do. That's what we do. We have eureka moments when we fish. Yeah, in the back.
I just wondered, what kind of results are they getting at Sloan Kettering?
Thank you. The question was, what kind of results are they getting at Sloan Kettering? They're in the phase 1 right now. And we don't know because that phase 1 hasn't been completed. So they're testing the first generation PSMA CAR T-cell in prostate cancer patients with late-stage metastatic disease. They should be getting close to finishing that because it's been almost a couple of years since they started it. So we should know, excuse me, very soon what kind of results they got. Now, remember, the phase 1 doesn't tell you anything about efficacy. It just tells you if the drug is safe. Because there's too few people in the trial to say that it worked or not. We have to wait to the phase 2, which is going to take another 2 to 3 years once they launch it. But that's a great question, very valid question. Yeah.
If this is being used on late-stage metastatic cancer, wouldn't it be that much more effective on early-stage cancer?
So the question is, wouldn't this be more effective on early-stage cancer rather than late-stage cancer? Why wait to the end of the game? So I agree. I think the sooner you can deploy something new and that you think has efficacy, the better. Unfortunately, the way that the clinical trial is set up is that this is an experimental therapy, right? It's never been given to people before. And so if you know already there is a therapy that is approved, that is safe, that has effectiveness, like hormone plus radiation, fracking, whatever that might be for you, you could be potentially giving an experimental therapy that could cause detriment, maybe even death, to that person, when you knew you could have given them something that does work and is safe. So the ethical consideration is that we need to try everything that is available, that is safe, before we can try something which we call as experimental, that we don't even know if it's safe. So once we establish that the CAR cells are safe, they're efficacious, they're better than or equivalent to the standard of care, then you can say, rather than try it in late-stage disease, we know it's safe, we know it works and efficacious in the late stage, let's move the bar. Let's treat the people who have intermediate prostate cancers, right? The Gleason 6s, the 7s. And then if that works, let's move it up to the people that have very early stage cancers, right? And that's how regulatory and ethical bodies want.
So that's many years before we get to that.
Many years. If you remember, well, you may not, but enzalutamide and abiraterone, when they first came on into the clinic, they were all tested in late stage disease. It worked very well. So now they've moved it earlier so that you can get it at the same time you're getting your radiation and your hormone, provided that your stage and your Gleason is all eligible for that treatment, right? And so that's kind of the progression of how things are going to come ethically and from a clinical trial standpoint. That's a great question, though.
Any more? Okay, we'd like to thank Dr. Lum.
Thank you very much.